Hello everyone, welcome to today's live broadcast, New Developments in Mass Spectrometry-Based Single-Cell Proteomics. I'm Chris Spivey, the Editorial Director of Biopharm International, and I'll be the moderator for today's event. We're very pleased to bring you this webcast presented by Biopharm International and sponsored by Thermo Fisher Scientific. I would like to share a brief statement from our sponsor. Thermo Fisher Scientific is a world leader in serving science. Its mission is to enable customers to make the world a healthier, cleaner, and safer place, whether their customers are accelerating life sciences research, solving complex analytical challenges, improving patient diagnostics and therapies, or increasing productivity in their laboratories. Thermo Fisher Scientific is there to support them. The company delivers an unrivaled combination of innovative technologies, purchasing convenience, and pharmaceutical services through their industry-leading brands Thermo Scientific, Applied Biosystems, Vitrogen, Fisher Scientific, Unity Lab Services, Patheon, and PPD. We have a few important announcements before we begin. This webcast is designed to be interactive, and we really encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark Help widget in the top right of your presentation window. Now it's my pleasure to introduce today's speakers. We're very pleased to be joined today by Karl Metzler. In the Metzler Lab, we are interested in developing new methods to increase the sensitivity, accuracy, and precision of protein identification, quantification, and the detection of post-translational modifications. Our aim is to use these optimized methods to answer fundamental biological questions. We currently aim to optimize miniaturized sample preparation workflows by employing and further adapting published techniques for various biological systems, organoids, tissue samples, and applications complementing sequences techniques with proteomic expression data. Furthermore, we investigate alternative labeling and acquisition strategies to enable accurate single-cell or other very low-input proteome measurements. Our second speaker, David Hadelmeyer, received his master's degree from the University of Natural Resources and Life Sciences, Vienna. As a master's student and technical assistant in the Metzler Lab at the Research Institute of Molecular Pathology, he co-developed and optimized the ProteoChip workflow for multiplexed single-cell proteomics. He is currently a proteomics application specialist at Selenion, Lyon, carrying out method development of single-cell proteomics workflows. And our last speaker, Manuel Matzinger, focuses on uncovering hidden protein-protein interaction networks and is keen to investigate the proteome of single cells with unprecedented sensitivity and depth. He is responsible for the coordination of the group's method development projects, grants, as well as in the supervision of students. As a trained chemist, his research focuses on the development of strategies to enrich low-abundant cross-linked peptides from complex matrices, enabling sufficient detection by means of mass spectrometry. Furthermore, his research focuses towards single-cell proteomics in tight collaboration with Selenion by improving all relevant aspects from sample prep to fine-tune chromatographic separation and MS acquisition with state-of-the-art equipment, a complete bundle of instrument parameters and tricks yielding in high proteome coverage and quantitative accuracy for ultra-low input is achieved. The optimized workflow strategies are applied aiming to investigate fetal brain on the most basic level using single cells from brain organoids. So thank you, everybody, for joining. Please, could we get started?
Ladies and gentlemen, I would like to thank you very much for attending our webinar entitled New Developments in Mass Spectrometry-based Single-Cell Proteomics. This webinar is held in cooperation with Selenium, who will present the latest development of the ZenOne. On this picture, you can have a quick look at our new mass spec facility, which is located in Vienna at the Vienna Bio Campus. It has recently been equipped with the latest generation of instruments, including thermoscientific Orbitrap Explorers for 80 mass spectrometer, thermoscientific Orbitrap Eclipse mass spectrometer, and thermoscientific Vanquish Neo HPLC system. It is one of the best equipped core facilities in Europe. Most single-cell studies focus on nucleic acids, especially the transcriptome, which represents all the expressed genes in the cell. Indeed, proteins are the worker bees of the cell. The amounts and the post-translational modifications, it's much closer to the phenotype. And that means that the disease diagnostic responds to drugs, all the human biology we want to inject, to control, steer, detect, it needs proteomics. I would like to take the liberty of making a comparison with a Formula One race. What is necessary to win a race? Is the most important component the racing car, the German racing driver, the Swiss constructor, or the financing from Austria? Our primary task in this project was to bring together the best components and to unite them into a beautiful symbiosis. Over multiple years of try and error, mostly error, we noticed for our overcells that in single-cell proteomics, every step needs to be optimized. From the efficient cell lysis, depending on the cell type, to analyze, to the enzymatic digest and the question of peptide labeling or not, towards chromatography separation, sensitive acquisition, and data analysis. Before a business project is carried out, a so-called reality check should always be performed. In this case, it's very important to ensure that the financial resources for such a project are to apply to the necessary financing in time. Regardless of the financial question, the scientific strategy of how I design experiments is just as important. I must say that there is currently no consensus in science, and we are in the so-called technical try and error phase. Here I would like to mention that many scientists have believed that single-cell proteomics does not work. One reason for this is the high dynamic range of proteins and the small sample size available. Therefore, to optimize your workflow, we like to work with a reference standard. Because they are small and large HeLa cells, and therefore the standard deviation with one cell population is very large. On the following example, I will show how we optimize the chromatography of 1 nanogram digested HeLa cell lysate. As chromatography material, we use a very nice new technology, so-called micropillars, which are now in the second generation. From this picture, it is easy to see the diameter of the second and the first generation. The second generation are now much smaller, which has massively increased the separation power. From this image, we can see a beautiful chromatogram of we have combined the best components that were available in 2021, namely the thermoscientific Orbitrap Exploris 480 mass spectrometer, the thermoscientific FAMES Pro unit, a new prototype of microbag columns, which is special designed for single-cell proteomics, and the thermoscientific protein discoverer software. In this example, we have used a second independent standard called K562 cell lysate instead of HeLa. In blue, you can see number of proteins, orange the number of peptides, in gray number of peptide spectral matches, and in yellow the total number of MS-MS spectra. All data were acquired without match between runs. Finally, we found approximately 2,000 proteins, less in only 0.5 nanogram human cell lysate, and almost 3,000 proteins in 1 nanogram human cell lysate. If you take a look of the distribution of the numbers, you see a flattering of the curve. This indicates that a 1CV method we can find as more than 3,600 proteins total. Here, finally, you see the evolution of sensitivity. We can meet our incredible target of increasing sensitivity by more than a factor of 10, starting with 10 nanogram HeLa and achieving better results with only 1 nanogram of human cell lysate 1 year later. What does it mean in reality when you talk to a thermo salesman? Explain to him that he should sell 10 times more Orbitrap mass spectrometers next year. Then he has reached the target for 2023.
Thank you all for joining us in this webinar. My name is David Hartelmeier. I'm a proteomics application specialist at Selenion, and I will introduce you to the ZenOne, which is the instrument used for all our single-cell proteomics workflows. The ZenOne is a pipetting robot that's capable of dispensing nanoliter volumes at high accuracy and with the ability of sorting cells while being temperature and humidity controlled. All these features are highly relevant in the field of single-cell proteomics, where you want to reduce your processing volumes as much as possible to minimize peptide adsorption to surfaces and to reduce the background noise in mass spectrometry measurements. Dispensing with the ZenOne is based on droplets. They are produced by a glass capillary. This capillary is surrounded by a piezoelectric ceramic, which induces acoustic waves when voltage is applied. This generates a droplet of a defined size depending on the voltage, pulse, and nozzle size, and usually has a volume of about 300 picoliter per drop. With selected parameters, the volume is very stable and has a CV of below 0.2%. The drop volume is determined, and depending on the volume that needs to be dispensed, a certain amount of droplets is released at 500 Hz. With this, both dispensing aqueous as well as organic solutions is possible. Cell isolation with the ZenOne is image-based. So 1 to 50 microliters of the sample is aspirated into the capillary, and droplets containing the cells are produced rather gently at 5 drops per second. Due to the low sample amount needed, this technology is perfectly suited for rare cells where the sample volume is limited. Cells can then be imaged with either bright field or with up to 4 fluorescence channels. And isolation can happen at 40 cells per minute. Now the cell sorting in a little bit more detail. The cell suspension is loaded into the glass capillary. The capillary is placed in front of the camera and 2 zones are defined in the capillary tip. The ejection zone in green is the volume of the next droplet that is ejected, and the sedimentation zone is the safety zone where it is checked that there is no other cell. After aspiration of the cell suspension, parameters like size, morphology, and fluorescence are checked and defined, followed by sorting of the cells. Only if there is a single cell in the ejection zone and no other cell in the sedimentation zone, a droplet is dispensed to the target. All other droplets are collected in the recovery vial directly below the camera. The images of the cells and the cell parameters are saved in a study folder and can afterwards be used for data analysis. In this video, you can see the cell sorting in live speed. The video on the bottom is the imaged capillary tip, which is tilted 90 degrees. You can see here that cells are only ejected to the target when exactly 1 cell is in the ejection zone. All other droplets that are produced are recovered in the vial below the camera, which can be filled with media or buffer solutions. As said, at the right concentration of cells, around 40 cells can be isolated per minute. This image-based cell isolation is capable of isolating single cells of a wide range of sizes and shapes. Using bright field imaging, cells between 3 and 80 micrometers can be isolated. In fluorescence mode, even smaller cells or particles are possible. In most cases, perfectly round cells are isolated. However, it is also no problem to isolate really elongated cells like cardiomyocytes. Sorting of cells can be done based on transmission or fluorescence. Isolation based on transmission has the advantage that it is faster at higher resolution and parameters like size and morphology can be chosen for sorting. However, with fluorescence, live dead, subpopulation sorting, and the isolation of rare cell types and small particles is possible. In close collaboration with the Mechler group, a multiplexed single-cell proteomics workflow was developed. Our workflow is very similar to a typical TMT experiment and consists of these 4 main steps. First, 100 nanoliters of master mix already containing protease, a mass-compatible lysis reagent, and buff is added to each vial. Cells are isolated into the master mix, followed by incubation for 2 hours at 50 degrees. Samples then are TMT labeled and quenched before pooling the cells together. Each chip contains 12 arrays of 16 vials preloaded with an oil to avoid evaporation during the process. Within one workflow, up to 3 chips can be used to process in parallel 576 cells. To pool the processed and labeled samples, the protochip is placed on top of the funnel part and quickly centrifuged. Within just 2 minutes of centrifugation, 36 sets are ready for measurements. The funnel part of the protochip containing the pooled samples then can be placed into a dedicated protochip holder for direct connection to the outer sampler. And in our first experiment, we just isolated HeLa cells. With this workflow, we identified on average close to 1,500 proteins per injection from a multiplexed set without a carrier channel, and up to 2,000 proteins when applying a 20x carrier channel. In the next experiment, we sorted similarly sized HeLa and HEK cells, resulting in comparable analysis depth. Based on 1,900 peptides, we were then able to cluster those two cell types and saw in our data respective proteome characteristics. Next, we did some single-cell analysis on in vitro derived cardioids by the Mendian group at IMBA Vienna. These cardioids are self-organizing into chamber-like structures and mimic the early cardiac development. We on average identified around 1,000 proteins per injection and were able to see the co-differentiation of the early cardiomyocyte and endothelial lineages in our data. This is visualized here by the PCA plot based on 1,100 peptides. So to conclude, the proteochip in combination with the ZenOne allows for high throughput single-cell proteomics, processing up to 576 cells in parallel. Here, the standard proteomics workflow is miniaturized and automated, eliminating all manual handling steps and introducing a centrifugation step to pool the samples, thereby enhancing the sensitivity of the single-cell analysis. And with that, I will hand over to Manuel. So if there are any questions, please let me know in the Q&A section of this webinar, and thank you for listening.
So my name is Manuel Matzinger. I'm a postdoctoral fellow in the Mechtler lab, and I had the chance to take over the work previously done on the proteochip in the group to further develop also a label-free alternative workflow. If you design a powerful proteomic workflow, you of course first think about needed ingredients. And the first ingredient that one thinks about is of course sample preparation. And I want to emphasize that sample preparation is really important, because if this fails, then everything else afterwards will fail as well. And especially when dealing with very low inputs, like this is the case in single-cell proteomics, you will face sufficient sample losses doing cleanup or when changing any tube. And this means that you would like to stay in a one-pot workflow setting. The next step, of course, is liquid chromatography in proteomics usually. And here you want to boost your separation power, you want to play around with gradient lengths, aiming to boost throughput, and you want to maximize loading capacity. For mass spectrometry, of course, you need to have a high sensitivity, high resolution device that is also very fast. You may want to add an additional separation dimension like ion mobility, aiming to reduce sample complexity. Plus, you can play around with different acquisition strategies like DDA or DIA. And last but not least, of course, data analysis. So here different tools are available, some are free, some not. And I will show you later that we prefer to work with chimeric spectra analysis and retention time prediction, as these yields in best results for us. A powerful workflow has also to deal with a huge dynamic range. So you can see here that the protein concentration within a single cell ranges across 7 orders of magnitude. In more complex environments like plasma, this can go up to even 10 orders of magnitude. Roughly 10,000 proteins, 10,000 different proteins are believed to be present within a cell at a single time point. And of those, the median copy number is only 18,000. And this is already rather close to the actual detection limit, and shows you that it's really hard to go deep into the proteome for a single cell analysis, because you will actually never identify proteins with low copy number in the cell. How difficult this is also depends on the protein content of your cell of interest. So smaller cells like HeLa will contain only roughly 200 picograms of protein, whereas the larger hepatocytes contain 600. And if you go for fibroblasts, for example, then it's even more, which makes your analytical problem a bit easier. To solve these problems, and this was already introduced before by Carl, we go for this advanced micropillar column chromatography system that allows us to work with high throughputs as the back pressure is very low, meaning we can do the loading and equilibration rather fast. And also the sample losses on the column are low as there is no porous material. And this column is within the Vanquish Neo system and connected to an Orbitrack Exploris interfaced with the FAMES Pro. For data analysis, we prefer to work with Proteome Discoverer and use the software Chimeris for the analysis of chimeric spectra, as mentioned before. So this does the identification for us, whereas label-free quantification is performed using IMP AP-Quant, which is an in-house developed tool. So how does Chimeris actually work? So I'd like to shortly introduce you to that. So the software makes first more or less conventional search, and with the peptides identified from that, it makes a calibration and uses its artificial intelligence to then do the second search, where it already knows the expected retention time of all theoretical peptides. And it can also do a spectral prediction. And by matching these predicted spectra to the actual ones, similar as you would do in a DRA run, you can really very nicely identify chimeric spectra. And this makes sense, as in usual DDA data, more than 40% of all spectra are actually chimeric. So in conclusion, when using this software, you can actually double the number of peptide identifications compared to a conventional search engine. So we played around with the isolation window of our DDA method. So we call this now wide window acquisition. And when opening up the isolation window, you create, of course, chimeric spectra on purpose. And since a single cell input sample is, of course, not that complex compared to a standard proteomic sample, you can actually open up this window rather wide to roughly 8 Thomson. And you can see in the left graph here that at 8 Thomson we reach saturation and maximum number of protein identifications from this sample. After that, it stays constant. However, the peptide level, the PSM level, is increased even more when broadening up the isolation window. And when you look to the right graph, you see that the resulting identification rate, so the number of peptide sequence matches per recorded MS2 spectrum, goes up to more than 80%. And we think this is quite impressive number for such a low input sample. So now we have already most of the ingredients, but of course we still need to do sample preparation. And this is our one-plot single-cell proteomic label-free workflow. After harvesting your cells, everything else is performed within the ZenOne that was introduced to you before by David. So this robot will dispense 1 microliter of a master mix already containing detergent and the protease to a 384-well plate. After that, individual cells are isolated per well and incubated for lysis and digestion. to hinder drying of the sample during digestion, we constantly add some water using the robot, keeping the volume at the same level. After that, we acidify to quench and supplement 5% DMSO to our samples to improve solubility of hydrophobic peptides and therefore the final recovery. The plate can then directly be placed into the autosampler of an LCMS and used for injection. So I would like to show you some of the evolution steps we did in this workflow. And the first step, of course, was to find optimal isolation parameters to really get only one individual cell per well. And you can see here when we started, so that's marked as day 1 here, with the standard parameters, only roughly 88% of the wells really contained one single cell, whereas the other had either no cell or multiple cells, or cells that were very irregularly shaped and therefore probably already dead. And when playing around with the diameter and elongation parameter for cell selection in the ZenOne, we can change this number to close to 100%. And this is, of course, a very important step towards full automatization of the workflow, because now we can trust that really each well contains one cell. And if you have this, second step is to play around with sample preparation parameters. So I mentioned before that we hydrate the sample to keep drying and that we add DMSO. So if you omit one of both, you will end up, as shown in this graph now, with roughly 500 proteins per cell. But if you combine both, so hinder drying and supplement DMSO, you can roughly double the number of identifications, both on protein and PSM level. In this when overlap, you can furthermore see that by adding DMSO to your workflow, you find more or less the same proteins than before, plus some 550 additional ones. So this shows to us that this is really a huge advantage and not just a random result. Another important step in developing this new workflow was, of course, to benchmark different data acquisition and data analysis algorithms against each other. So you can see in this example here a DDA1 that was either analyzed using Chimeris that I explained to you before, or the conventional search engine Amanda that was developed by our group back in 2014. So Amanda is also able to deal with chimeric spectra, but it has no artificial intelligence-based spectral prediction. And you see very clearly the advantage of that, which again boosts your numbers roughly by factor 2. If throughput, on the other hand, matters, then we need, of course, to shorten the gradient. And the idea of MS1 search pi, which is the yellow bar, is that we really omit any MS2-based fragmentation. We record only MS1 spectra, and by that, of course, save a lot of time for the instrument and can go down to rather short gradients. And you can see that with 1/3 of the gradient length, we still identify the same number of proteins than before with Amanda. When you again look into the when overlap of the identified proteins, you see that Chimeris identifies the exact same proteins than Amanda plus some additional ones, which gives us again confidence in the correctness of the new data. Whereas with MS1 search pi, this overlap is not 100%, but still roughly 70%, which is a similar number as published for that software. And last but not least, of course, you need to be impressed by your results. And we were quite impressed by the dynamic range that we reached. So this is the data again from one individual cell, and we were able to cover up to 5 orders of magnitude within that, which is not the whole range that I showed to you before, but already quite deep for that low input amount. So to sum this up, we now have a rather loss-free sample preparation with an improved recovery by storage in the MSO, where we hinder any drying by working under high relative humidity and by automated supplementation of water. We boosted our ID numbers by intelligent data analysis and the throughput by short gradients. And these short gradients also will improve your sensitivity thanks to sharper and therefore higher peaks. So to go back to the ingredients, we now have all of them together. For sample preparation, we prefer to use the ZenOne and work only in one well from the beginning to the end of the whole workflow. For chromatography, these micropillar columns showed the best performance in our hands. Regarding mass spectrometry, this wide window acquisition producing chimeric spectra on purpose showed excellent results for us, or of course VRA is another option to go. And for data analysis, Chimeris outperforms all other competitors. So with this, I'd like to summarize the content of this whole webinar. The combination of advanced chromatography, wide window acquisition, and Chimeris allowed us to reach outstanding sensitivity and cover a very wide dynamic range. For standard proteomics samples, this yields in more than 10,000 proteins per injection. For individual cells, working with a label-free workflow without doing any match between runs or adding any carrier, this yields in more than 1,000 proteins per cell. And when going for multiplexing, working with the Proteo chip, this yields in 1,500 proteins per run without the use of any carrier channel. So I'd like to end this webinar by summarizing what future challenges and limitations we see for the field of single-cell proteomics. So most researchers in the field would agree that to really have a solid statistics and investigate cellular heterogeneity in biological samples, we really need to boost up the number of cells being analyzed, and a good number would be more than 1,000 per day. So this is something hard to reach at the moment, but with even higher level multiplexing and or very short gradients, we are at least on the right way to go there. Another thing, of course, that is problematic even more with higher number of replicates is that you will run into missing values that will hamper proper quantification across replicates. And last but not least, of course, sensitivity is still limiting, and here hard and software needs to be improved in future to further boost this. So what are limitations? So the most important one might be money in the end. So this was already mentioned before by Carl. A robot to do sample preparation in a miniaturized layout will cost you 250,000 euros. A mass spec might be even more expensive, at least if it's a good one. And also consumables like columns or reagents for multiplexing, as well as software, will cost a lot of money. And finally, you need know-how. So you need to figure out which workflow works best. And this is really dependent on the sample type and on the questions you ask in your project. So you really need to figure this out for every project again. And for that, you need well-trained staff and time. And as you all know, time is again money. So with this, we would like to thank our colleagues at Thermo Fisher Scientific, and especially Jeff did most of the development work, developing the micropillar columns. And collaborates very tightly with us here at the Campus Vienna Bio Center, and also the team in Germering, especially Hongzheng and Alexander, who worked a lot together with us solving chromatographic issues. And last but not least, our proteomics core facility here and on campus for this very inspiring working environment. And especially Elisabeth, a former master's student of mine who did most of the work for the label-free workflow, as well as Claudia and David, both already moved forward in their careers now, but they were the main drivers for the development of the Proteo chip. Yeah, and last but not least, thank you for your attention, and we're now open to all questions.
Well, thank you, David, Carl, and Manuel, for such informative presentations. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found directly below the video player. And because of the large audience size today, we're not going to be able to get to all the questions, but never fear, we'll be able to answer all of the questions offline. So the first question that I see is for Carl. And Carl, someone is interested in the peak width using the pharmafluidics version 2 columns. Could you elaborate a little on that?
Hello, everybody. Good morning, good evening. With the generation 2 of the pharmafluidics columns, we have a peak width approximately, peak half width approximately 3 to 4 seconds. And the big advantages to the generation 1 are the smaller pillar size, the robustness of the columns. At the beginning of the column and end of the column, there's a frit. And also it's equipped with nanovipers.
Okay, thank you. The next question actually is probably also for you, Carl. Please, can you provide more details about the WWA method?
The WWA method, it's quite new in the field of proteomics, and it was published recently by my co-workers, and one of sitting here, Manuel. In principle, it's you open the quad, dependent on the sample amount, up to isolation width of 10 or 20 Dalton, and you collect as much as you can on chimeric spectra. In old days, it was quite difficult to handle this chimeric spectra. But new in the Protein Discover software 3.0, there's a new algorithm included. It's called Chimeris, which runs in the Amazon cloud. And with Chimeris, you can handle up to 10 PSMs per MSM spectra. And therefore, it's a really powerful tool to get really high numbers of proteins or peptides out.
Terrific. Thank you. I appreciate that. The next question, I believe, is more for David. How can I determine my cell-specific parameters? And they've listed size, morphology, and fluorescence as examples.
So when the cell is imaged by the ZenOne, the image of the cell is compared to the background, and the program automatically determines the diameter of the cell, the circularity, the elongation, all these factors, and then decides if the cell is sorted to the target or recovered within a recovery vial. And all these parameters then are automatically saved within one Excel sheet that can then be used for data analysis afterwards.
I understand. Thank you. Good explanation. The next question, why PCA analysis on peptides and not protein groups, David?
So yeah, we really wanted to eliminate all biases that are introduced by grouping the peptides together and by normalizing the peptides to protein groups. So that's the reason why we did our PCA plots based on the peptide levels.
Okay, so sticking with you, I wonder if you have any suggestion for configuring autosampler for direct injection for proteome chip funnel on the LC systems other than U3000.
Yes, that's true. So for the moment, only the U3000 can be used for direct injection. However, we are working very close together with Thermo Fisher Scientific to also make it possible for the other systems, like the Vanquish Neo. And yeah, that is still work in progress, but will be for sure also be possible in the near future.
Okay, so while you're on a roll, can the tandem mass tag plexing rate be increased, and how would you increase it?
So the current maximum amount of TMT labels available is 18 plex. Our chip was developed in a phase where still the TMT Pro was limited to 16 labels. And we are also currently working on a prototype chip that makes higher plexing possible. But for the moment, our current prototype chip is limited to 16 plex. However, you could also use one of the other additional plexes of the 18 plex to spike in a carrier or reference channel, if that is needed.
Terrific. Thank you. That does make sense. So switching to Manuel, what role is DMSO playing here?
Well, that's an excellent question, of course. We played around with a lot of different possibilities to minimize all possible sample losses, and then found that DMSO seems to keep, especially the hydrophobic peptides, solubilized, and therefore also after storage of the sample in the freezer and also within the autosampler, the recovery in the end is much higher, and eventually also the peptide numbers that we obtain.
Thank you. Okay. So we're getting more questions. I stick with you, Manuel. How are the samples pulled and the proteome chip transferred to mass spec vials to ensure there is no sample loss?
Yeah, so as mentioned before in the presentation of David, there are in the end two possibilities. You can either directly inject out the proteome chip, meaning no sample transfer, which I guess is the ideal scenario. But at the moment, this is only compatible with the 1000, as answered in the previous question. If you want to transfer, then a good idea might be to try to cover the wire you use. So what, to the best of my knowledge, David did when he was here was to cover PCR tubes with polyethylene glycol, and with this also lowered the absorption of the peptides to the balls with this managed injection.
David, would you like to add anything to that?
No, I think that was perfectly answered by Manuel. Thank you
for
answering this question.
Yeah, they're not easy to answer on the fly. So I'll go to Manuel still then. So did you compare between the 384 well plate and proteome chip processing in terms of protein peptide identification, and what was the differences that you found?
We did not directly compare. Of course, the proteome chip is more meant for multiplexing, whereas our 384 well plate approach is more meant for very free work. So in the multiplexing, you of course have in the end up to 16 cells or whatever your multiplexing factor. This gives you slightly more IDs. If you do not do any sample transfer, then the final protein ID numbers is, I guess, comparable.
Okay, thank you. Okay, so the next question, I believe, is appropriate for David. So back to the tandem mass spec tagging. What is the ratio of TMT to protein in cell one? Does the TMT byproducts impact on the protein ID? It's a complicated question. And what's the protein concentration range in cell one? So maybe take it bit by bit.
Yeah, so if I answered the question right, so we dilute the TMT reagents quite a bit. So it's a 1:8 dilution of the original concentration. And then we only use 100 nanoliters of that dilution. And with that, we really manage to minimize the background noise of unreacted TMT and to not overlabel our peptides. So when you think about a single cell is about 0.5 nanogram of peptides, and then obviously we don't reach the recommended peptide to TMT label ratio for bulk, but that's just not feasible in this single cell proteomics amount.
Yeah. Now, these questions are flooding in. The next one, Carl, is actually a very good question, I believe. Any pointers on the best way to harvest the cells to put into the cell sorter?
I think it's a question for David.
Ah, David.
Sorry, could you repeat the question? I couldn't hear.
Yeah, do you have any pointers for the best way for cell harvesting to put into the cell sorter?
Yes, so what we usually do is using trypsin for harvesting the cells, but of course then all the or many of the membrane proteins might be also cleaved in the process. So what can also be done is just scraping the cells from the plates, but usually what we did in all our workflows was using the trypsin method for that.
Okay, thank you. So I'm scrambling to keep up with the flow of the questions. Carl, I think this one hopefully is more in your ballpark. How well is the Chimera adapted to study PTMs?
That's an excellent question. Chimeras can at the moment handle methane oxidation and any kind of TMT. Indeed, in this version, Chimeras is not able to handle phosphorylation, but I hope that this was soon adapted.
Okay. So Manuel, we have another question for you. Why does DMSO increase recovery or sensitivity?
Well, we think that DMSO keeps these, especially hydrophobic peptides, better in solution, as it's a organic solvent versus water, right? And this is exactly the reason why adsorptive losses are minimized and therefore recovery is improved.
Okay. So the next question for David, I believe. How do you verify that single cell is dispensed per well in cell 1?
So as in my presentation there, the tip of the capillary is divided in two zones: the ejection zone, where you know, okay, this will be the next droplet that will be produced, and we introduce also a safety zone. So when you start with a cell solution, you first do a mapping to really see, okay, how many droplets are needed in order to dispense a single cell, and the program then evaluates how much one droplet is, and with that we can also image, like, after the cell the next droplet, and so we can see, okay, there was only one droplet containing one cell and not two cells.
Perfect. So next question, two-parter for David. I believe it's for David. What is the role of the carrier of the SC mass spec, and can you give an example?
So the carrier was introduced Nikolai Slavov in 2018, and the carrier really is here in order to reduce all peptide losses within the workflow due to peptide adsorption and in the chromatography. And when using TMT, usually this ratio of single cells to carrier might also matter quite a bit. So what we did was trying to reduce this carrier ratio as much as possible in order to not influence the single cell quantification too much.
I see. Thank you. For Carl, I have a question. How did you minimize the post-column diffusion after the LC separation using the micropillar column separation?
The post-column fusion was a problem with the generation 1, but it was difficult to connect the column to the needle of the inlet of the mass spec. It's not much more now easier with generation 2, where we can handle the connection with nanovipers. And if you have broader peaks, it means you have dead volumes, and you have to improve your dead volume to get sharper peaks and play with the settings.
Okay, thank you. That's very helpful. David, I believe this is for you. How does cell 1 differentiate two morphologically similar cells? And a follow-up, can we choose to select one type visually, or what options really do you have?
So with the possibility of fluorescence in the ZenOne, obviously your cells could be fluorescently tagged. So that would be one way how to differentiate those cells. But yeah, other than that, you can distinguish the cells by cell size and elongation. And based on those, the cells could be differentiated in addition to the fluorescence.
Okay, so you guys are handling these quickly. We're running out of time, but there's time for a few more. Carl, will Chimeras handle other cysteine alkylation reagents, for example, MMTS or IAA?
Oh, that's a great question. That's also something missing in Chimeras, and we will hope that we'll include it in the next release soon.
Okay. So we've answered quite a few of these questions. I'm looking for other interesting ones just as the time does wind down. This is for Manuel. How do you handle the dynamic exclusion with the 8th selection window?
So that's, of course, a very good question. So indeed, we use only a dynamic exclusion window around the actual precursor mass and not over the entire 8 Thomson range, but instead use a rather long dynamic exclusion time of 120 seconds. Yeah, that's all.
Okay. Thank you. That does answer. That was a more complicated question, but you got to the heart of that fairly quickly. Well, actually, I'm afraid I think that we have run out of time, unless there's one more question that I'm sort of reluctant to pass up the good questions on the live. But people have been sending these in, and when they look at the webcast as a static thing, they can also send in questions. So you guys are going to be busy for the next week or two answering these questions. So I will thank you, everyone, for such great presentations and for giving such good answers to these questions. As I said, all questions will be answered offline that have come in. We got a bunch. Unfortunately, we're out of time, but I want to thank the audience for attending and participating in today's event. And I'd also like to thank our sponsor, Thermo Fisher Scientific, for making today's educational webcast possible. We'd like to ask everyone in the audience to participate in a brief survey. The survey will populate automatically after today's presentation has ended. You'll receive an email alerting you when this webcast will be available for replay, and we invite you to forward that announcement to your colleagues who may have missed today's live event. So David, Carl, Manuel, thank you very, very much for giving your time and for giving your insights. Thanks to everybody for joining us today, and we'll see you next time. Thank you and goodbye.